Welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PI Collaborative Community aims to harmonize approaches to speed delivery to patients through collaboration in the pre-competitive space. The PI-CC is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law, because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection, since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda, and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of a discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward.
This meeting is being recorded.
All right, so welcome to Seer 2.0. Let's do a brief round of introductions. Vivian, do you want to start?
Sure. My name is Vivian Sintron, and I work for Agilent Technologies. I am the global head for pharma partnerships and engagements, and also very interested on cell-free DNA. That is one of the aspects that Agilent is looking very closely.
Perfect. Uta?
Yeah, I'm Uta Schmidt-Straßburger. I'm from Ulm University. I'm currently directing scientifically a study program that covers, among others, also the issues, topics that you're interested in. And Ahmad is a former student of ours, now a lecturer. So I think I'll just elegantly hand over to him.
Yeah, I'm an ophthalmologist, ocular oncologist, and interested in epidemiology. I'm working recently in Berlin and Gießen in Germany, formerly Ulm. And yeah, this year is one of my very close interests. Yeah.
Perfect. Thank you. Emma? Can't hear you.
Try to choose the right microphone from your audio options.
Better?
Yeah.
Fantastic. Sorry about that. I'm Emma. I work in CID with Joe, and I don't know what to say. I'm an undergraduate student I'm a data analyst. I have learned a lot about Seer and really think it's a fascinating data registry, and I'm very excited for where this project's going to go.
Perfect. I'm Joe. We do a lot of tech assessments, and together with Ulla, who's joining momentarily, we lead this Pathology Innovation Collaborative Community and have been working on this project for a while. So maybe before we jump to Emma's presentation to outline this, there's like 2 or 3 things to as a concept. So first of all, the basic premise of this is to use the Seer database to determine the number of cancer cases diagnosed using the light microscope. And while that is a fair question, and it sounds a little bit bizarre, but that has apparently not been answered yet. So this is providing literally 1%, so something like 90 or 95% or 85%, but we will figure that out later. The reason for that is that there are two main sort of new technologies entering the market. One of that is cell-free DNA testing. The other one is digital pathology and AI. And so before coming to this sort of point of why those technologies are important, several groups have asked within PyCC whether we could start a cell-free DNA work group and or a project about various regulatory aspects. And in relation to that, recently the FDA issued a guidance document on how to do early cancer detection using cell-free DNA. We can share this with the group. Of course, we put it on the website as well. And that provides some hints that we should definitely incorporate into this project because it actually changes the way we diagnose cancer. So that will change, of course, then the modality by which cancer is diagnosed. And then the second part, digital pathology and AI, is probably still using the same thing like a light microscope and light microscopic applications, but it's a new technology added on top of that, similar to, let's say, formerly immunohistochemistry or flow cytometry or molecular diagnostics. So these are, I would call it like additional tools and modifications. So again, since we're mostly focusing on regulatory aspects, the first AI algorithm, in this case for prostate cancer detection, has been approved. And we're soon hosting a session to go through the decision summary of that particular, let's call it, decision of approval for this. It's from a company called Page Prostate, but we want to go through this to see what lessons can be derived from that approval to account for in this project. So simply put, the Seer project as it existed originally is extended on the one hand by a work group and a project with cell-free DNA, where we're going to ask many interested parties to provide that as a new trend. And then the other part, which is a decision summary from, the Page Prostate, to just illustrate in one specific cancer how that may actually change the tracking and tracing of the epidemiologic factors. So with Vivian, who's very interested in cell-free, and Uta and Ahmad with their expertise, I think some of the lessons that we can learn from that from an epidemiologic perspective or more global health perspective, I think it will be very interesting to hear what you're thinking. So that's just a very quick overview about the, let's say, new aspects. Does anyone have questions about that or initial comments? Vivian, you're very interested in cell-free, correct?
Yes, I am. So this is a space that is of heavy interest at the present time. As most of you will know, the oncologist requires to do a tissue biopsy, and they will embed that tissue in parafilm, and that tissue goes into a laboratory and it's being viewed under the microscope, under the tools that you explained, Dr. Lenners. However, it could be a good practice, and some oncologists are doing this already, where they are at the same time sending another sample, blood sample, to a laboratory. And this blood sample will address different kind of markers in the interest of the potential cancer that they are looking into. So those are the areas. How, if this could be a parallel world, what if this will be the parallel world that will continue, or if this will be the future, or do we have enough data, global data, on the upcoming clinical trials from the different companies, pharma companies, to know if this by itself could be the initial step instead of doing a biopsy.
I'm just typing this, but Ahmad, could you comment on this, because this this concurrent, or what you described as parallel world, is that something that is known in epidemiology and how to account for it? Because that's basically multiple data points for one time point.
Sure, of course. For example, in non-cell small cancer, this is a cancer that typically will get biopsy, and if at the same time would be some kind of specific marker that can be also done as blood, that it's something that it's being done by some oncologists. I will say maybe 50% of the oncologists in the European nation are doing this. A little bit maybe late stage for US. They depend in the US depend more on the tissue, and then meanwhile they wait for the tissue 14 days or 16 days. Now they are learning that they could have been draw another blood tube EDTA that can be sent to a specific laboratory for the specific biomarker that it's being looked in here. Not all obviously we are looking for solid tumors. So this might be the initial space into solid tumors. In Europe, it is becoming very trendy to start looking into this parallel opportunity where you can have the initial blood drawn, and then trying to identify the biomarkers. Different solid cancers, not only non-small cell lung cancer, but cancers that can be shedding into the blood, and that can stand immediate transportation to do these immediate testing. So that it's something that can be, it could be a great opportunity.
Uta, I see you raised your hand.
Yes, I did. I would like to just point out that it might seem that almost everyone is doing that in Europe, but my perception of our reality is somewhat different. So it is something that is very appealing to the larger comprehensive cancer centers, but it is not in widespread clinical use in a conventional outpatient clinic. So I would be very careful with this projection, because right now there are initial clinical trials ongoing, but they are subject to having negotiation, particularly concerning the reimbursement of all the efforts that go in there. And it is really more of a pioneering work in comprehensive cancer centers, but it is not something that is widespread use as of now. And right now, my perception is also that depending on in Germany at least, depending on the different tumor entities, the practice will be different. So I think in breast, we might be at an advantage. But for instance, with regard to other tumor entities, I would be more reluctant to shout out a grade A as of now.
Ahmad, can I ask you again, please, to comment on this sort of dual?
Yeah, I know such issue, but the point is, in Seer you get one thing. So you have one option to know the pathological confirmation, and it is the highest method that the diagnosis reaches with. So if the diagnosis was confirmed using clinical diagnosis and pathological diagnosis in the same time, the only thing you will get is the pathological diagnosis. If you get it, if you get it by radiological diagnosis and at the same time pathological diagnosis, then you get one thing is which is the pathological diagnosis. So they give you one confirmation, one method of confirmation, and you don't know if other methods were used for the diagnosis.
Yeah, it's interesting because, one of the possible outcomes here is that we could at least discuss advocating for a finer granularity in terms of tracking this, especially when, when parsing this out by individual tumor types. For some cancers, a tissue sample is just not feasible, or, some of the peripheral blood tests now even distinguish what the cancer of unknown primary actually is, or make some good, reasonable assumptions about it. But I don't know whether they can be accurately reflected in the diagnostic confirmation codes as they exist today. So no, we don't have a lot of time, but Emma has prepared a couple of slides. So what I would kindly ask you to do, now that we briefly discussed the various vantage points that we want to tackle this problem, if you could make notes on, important aspects that may not be as clear, because with a project like this, it's very, very important to have a very good setting of the stage. And we wanted to use this to go over this. It's just a few slides, but if you can take notes and then just be very critical, as I know you all love to, and just so we have a very good starting point. That's the main aim for this meeting, and then we can maybe go over your notes, and yeah, that would be great. Emma?
Here we go. So I hope everyone can see this little PowerPoint. Ula put together this great introductory image for Seer 2.0. And I think as Joe kind of mentioned, our big point is that we're looking to go into the future trends and not just provide a baseline. And so with that, our key question, as we've been talking about, is kind of to what degree do pathologists depend on light microscopy for cancer diagnosis? As we said, there's really no clear answer in the literature yet. And with all of these rapid development of technologies, we would like to set that baseline to know where, to know where the field is off to. With that in mind, our primary endpoint is, as Joe said, to get one number for the percentage of cancer diagnoses by light microscopy, with the secondary endpoints of can we break that percentage down by ICDO code, by the primary site, and by the state of patient residence. These secondary endpoints are not in stone, and we absolutely welcome any ideas for other endpoints. Our basic data overview from Seer, as you all probably know, is that when a patient comes in, they will receive a cancer diagnosis. That diagnosis will go to the local registry, then to the state registry, which will then go to the Seer data set with the addition of removing duplicates and consolidating the cases. And Seer as a whole is only a registry that looks at the US population. And I know initially we had talked briefly about including some NACER numbers as well, which covers all of North America, which I think I would be interested in continuing that conversation, as I feel like we could probably cover more, seeing that Seer really only covers about 35% of the US population. But yes, so this is Seer. We query it on the Seer stat data set. And what we're looking for in diagnostic confirmation is it to be in one of the 4 top categories of positive histology, cytology, immunophenotyping or positive genetic study, or not specified but microscopically confirmed method. These are what we are classifying as diagnosed by microscopy, whereas 5 through 8 is non-diagnostic non-microscopic diagnosis. And then 9 is unknown, which we are mainly keeping in its own category. With that, this is roughly what our data looks like, keeping the same headings on the as above. Oh, sorry. I'm seeing Vivian's comment now. Does the Seer cover patients that are also on current clinical trials? I'm not entirely sure on that. I'm not sure if anyone else knows, but I will definitely figure that out and get back to you.
I do not think so. I think that is so that are on the current clinical trials would require real-time tracking, and I don't think that that's possible. So Seer is, of course, a multi-year database where the curation usually is a year to 3 years behind current state. I'm not sure whether there are fields whether the patient has been treated in a trial. That may be possible in some of the later years, but I'm not too familiar with all the supplemental components. But that's a good point.
So I'm sorry, what was the question?
Whether Seer covers whether a patient is currently and or was in a clinical trial.
So maybe there is data behind the scenes that we call custom data, but it is not in the public data set. So this point is not addressed in the public data set available to access. But you can ask the Seer team if they can provide such field or not.
Yes, sometimes this is a question. Sometimes during the clinical trials, there is the interim review of the trial that is public or might be public on clinicaltrials.gov, at least in the United States, and can have a little bit of that information. But again, I don't know too much detail.
Yeah, so sorry about that. We will I will reach out to the Seer team and see if we can get some more details about if patients are included. Sorry about that. If patients are included, and if we can get any numbers about more recent years. But generally, this is what our data looks like. We've separated it into solid tumors and heme tumors. Here are the ranges for ICDO. Solid tumors go from 8,000 to 9,589, whereas 9,900 begins heme tumors. Of note here, 1 through 4 represents microscopically diagnosed, except X3, which, as we can see, is positive histology and immunophenotyping and or a positive genetic study, is not a relevant diagnostic category for solid tumors. However, we do see, as in this primitive neurodermal tumor, in that they are often diagnosed. I'm unsure. They are sometimes diagnosed as this category of 3, which I'm not entirely sure how to handle yet. But that is something that we can also talk about, because they're not often seen, but they are seen throughout. And then we can take all of these, all of the data across ICDO codes and conglomerate total cases and percent diagnosed microscopically. Here's a random sampling of our data, and you can see percentage diagnosed microscopically ranges from 100% to, in this case, down to 87. But I believe our actual percent diagnosed microscopically, our lowest range is much, much lower, though I'm not sure what it is off the top of my head. I apologize. You can see a similar thing for heme tumors. though overall the percentage diagnosed microscopically is significantly lower, and there are many more tumors that are diagnosed non-microscopically, which is a bit interesting. And X3 is a category here that is a viable diagnostic confirmation. And then in Seer, in the second generation of Seer, Seer 2.0, what were our main new components is the inclusion of cell-free DNA advancements as we begin with, while considering including commercial partners and their data, the inclusion of digital pathology and where we can get some data to support that argument. And we have a question of the inclusion of a Seer database update. Over the course of this project, the Seer database, we did gain an additional year of knowledge. The year of 2019, the data was just released the middle of April. And so we were able to redo some of our analyses using this new data. And then we're also just interested in these other new trends and what you all are kind of seeing around and what you think is going to change how tumors are diagnosed in the coming years. That's all I have in slides, but I am welcome to take any questions or.
Emma, can you go back to the classifiers, so the diagnostic confirmation codes? So, one element that I just noticed is the, X5 positive laboratory test. So I don't know, coders are usually very well acquainted with the definitions, but some people may consider, let's say blood work or, let's say work on a hemocytometer, possibly a positive laboratory test, but that may actually entail microscopic examination. So I'm not sure, but I think the subcategorization of these diagnostic confirmation codes is of course taken from NACER, so I think that's good. I just made that note related to possible limitations with the coding here. Other comments? Ahmed, Vivian, Uta.
So the Seer 2.0 is a proposal or something that is already running?
This is the project that we started, but we wanted to incorporate, other data elements like cell-free, as well as, digital pathology, AI. So we currently have not conceptualized in this slide deck how we would obtain, for example, adoption data of some sort. As Uta mentioned, it cannot yet be considered standard of care. We know that there's numerous commercial vendors, some represented on the call, that work in this space. They probably have their data. So, one thing that we considered is, as part of this 2.0 of this newer project, to reach out to some vendors and ask whether they would share sort of adoption numbers of tests run over time. And as long as we're not restricting it to a single vendor, we may not be able to approach national level, but we could at least say, for example, across the 10 vendors that participated, we see the following adoption over the last, 3, 4, 5 years. Now, of course, the Seer database doesn't go all the way to 2022. So what was the most recent year, Emma, that we had?
We have until 2019.
Yeah. So it's we may end with 2019 with Seer and then project onwards with cell-free. But I think this is probably the right time to discuss some of the limitations of the current approach. But I think just the percentage is a little weak, if you know what I mean. So we wanted to make sure that it's clear what trends are, what the field is being facing right now is probably the best way of putting that.
Dr. Lenners and Emma, have you approached also networks that do clinical trials? Sometimes when the authorship of the clinical trial is obviously getting the budget from pharma to produce a clinical trial, they could potentially embed this kind of scenario or partner with you on that specific trial. Have you approached that kind of network or consortium groups?
No. I think the focus for now was mostly routine diagnostics, not clinical trials. I mean, that was sort of the... Do you have someone in mind?
In the past, I worked for a consortium, a network, and it is a network that is actually pursuing cell-free DNA with other opportunities, not Agilent, but with other opportunities that are in the market specifically to have an understanding if in fact the tumor is shedding, how much is shedding, what is the time lapse of shedding, and then what will be the pharmaco indication, if it's going to be a dual pharmaco or one single pharmaco after radiation. So I have one in mind and I can introduce you to that one in particular. They operate in different sections or segments, for example, thoracic or GI or GU. So different kind of segments. You might want to maybe start with one segment that at least you know will be shedding into cell-free DNA, and then present the idea to the chair of that committee. And hopefully that partnership will get you into tons of patients right now that are more than routine. So they are utilizing perhaps dual the testing, meaning the microscopy for the FDP, and then also the cell-free DNA.
That sounds great.
Okay, so I will set up a time for that pursue if it is on your interest.
Yeah, if it's more cell-free related, it may be good to either use this to supplement our data here, but let's just keep an open mind that there may be other groups that will join the cell-free, let's say, focus.
Yes.
Yep.
I have some ideas or comments. So what do you mean by first the 2.0 or Seer 2.0?
An updated project.
Okay, another version of it. So I thought in the beginning, because you know, from the internet 1.0 to internet 2.0, the difference was that internet or web 2.0 is the interaction or the interactivity or the interaction between the people, or the ability to network between the people over the internet. And the third, or what they call Web 3.0, is the Internet of Things. The first impression was that Seer 2.0 will allow the crowdsourcing of the efforts in the Seer. So instead of just getting the data from the cancer registries and curating them, this will, I mean, integrating digital pathology and multiple sources together and making the data not open source, but somehow open source will allow people to provide more ideas, more efforts to for example, the pathological diagnosis. One of the examples that one of my students have published recently, former students, he is now a PhD. I mean, he's was graduated from Emory, and one of his projects was the crowdsourcing of the pathology slides. I mean, the analysis of the pathology slides were done by tens of the other physicians. So to get a reliable diagnosis. So maybe by integrating the digital pathology to the Seer results, this will allow the people to reach better diagnoses, and also will allow researchers or will allow the developers to develop tools that will improve the performance of cancer registries.
Yeah, that's a great suggestion. So quickly the first part, we would like to, just let's call it solicit crowdsourced input for cell-free, because there is no such thing as a national database for, let's call it, adoption of cell-free DNA. So the data will definitely be biased, and we may not be able to include it kind of like one to one, but it's more like two independent things. For example, if we plot the data that Emma has curated over time, it's our assumption that that should stay relatively consistent. While contrasting that with cell-free data, there's clearly an uptick in cell-free utilization. So it's more sort of a conceptual contrasting rather than a one-by-one comparison. The second part, Ahmad, what you mentioned is phenomenal. I mean, if there would be the option to, let's say, have broad-scale digital pathology and include, let's say, representative or diagnostic fields or slides as digital pathology data sources with these publicly available data sets, that of course would enable a completely new realm in research. And of course, AI would probably play a role. And then of course the question is, would that data be considered regulatory grade, or, what would be the governance structure that needs to be attached to that to make sure that the right slide is attached to the right patient and is actually accurately reflecting what the diagnostic confirmation is. I very much like that. That is really, really good.
Thank you.
Uta, other feedback? Or Vivian, other?
Well, just as I signaled already, I think right now the coverage of patients for whom cell-free DNA was analyzed might not be very high. So the beauty of both the Seer and the NACER is that they are rather representative, whereas, at least in my perception, patients for whom cell-free DNA was analyzed are not, or are rather few, at least here in but I do have a little bit of insight. So I do not know how the situation is currently in the US in a standard hospital. Could you elaborate on that, Joe?
So I would say the test is mostly done by commercial vendors, and the ability to ship this out and track it and use it appropriately, I think, is in a in a largely oversimplified way distributed as well. Sort of some people use it for one specific, let's call it, monitoring purpose, while others use it when they cannot easily obtain a biopsy. And I presume what you describe is sort of a gradient between academic medical center and then tertiary, secondary to community practices. I'm not sure whether we could I mean, that is, Vivian, for the cell-free group, those would be the key questions to see what that looks like. Now, at the same time, the fact that the FDA is issuing a guidance document for cell-free testing in early onset, meaning basically for screening, suggests that is, of course, there's a certain risk attached to it. I mean, we all know the Theranos story and what the issues were with that, but I think early cancer screening using peripheral blood is definitely coming. So the guidance document is definitely providing some key outline of what that would look like. Let me just actually share this in the chat so everyone has it. So we should, but it's heterogeneous, just like you described. I presume at the national scale in the US, it sounds pretty much identical to what you described, Uta, in Germany.
There are some big centers here in the US that are utilizing the tumor boards, and this is very much interactive, going through slides of patients and also combining it with cell-free DNA for these are more difficult diagnosis of patients, or patients that these are very key cases. So my question is, or my comment will be, getting into those tumor boards or participating, it's very limited, and it's only for residents or MDs that are part of those centers. But maybe that kind of association will be a key here, just to, because again, this is trying to utilize some of the dual tools that we have at hand, either, well, the slide, also at the same time the cell-free, and it will be very interactive, as Ahmad was pointing out. So I'm wondering about that kind of opportunity. But again, they are very limited in numbers. What you can go through the tumor boards.
Yeah, the issue is to get from let's say, a monitoring of a patient to the utilization at a, let's call it, a cross-patient level. That's kind of like the ultimate crux of epidemiology, right? That's very tough. So we will have to find a balance. But one element regarding just self-free utilization, I'm sure there are some papers out there on surveys. What people prefer. For example, I'm not even sure if I don't know, a certain lesion can't be biopsied, whether every provider across the various settings is thinking about the option even right now. It's kind of like exposure driven probably, but that depends, of course, on the setting and, the knowledge of the provider.
Right.
So unfortunately, I have to run to the next meeting. I think you may have seen on Friday, we wanted to look at the time trends, meaning what we have by then. And, Emma is putting together a few figures. And then we can discuss sort of like what we can derive from that. And then, we can, if you have other contacts for like, cell-free companies or so, we can probably reach out and then see what we can do. Yeah, so if you know other people, I think Uta, I mentioned, if Raquel is interested in this, right? She's working on this. There's probably a little more interest in, if there's like some new technology included. All right.
That's great.
Thank you very much for this valuable input, and we will incorporate, and then the material as well as this recording and the slide deck will be up on the website. And I hope to see you on Friday.
Great. Thank you. Have a great day.
Bye-bye.
Bye.
Yeah.